Um, what I want to talk to you is a story about three things. One is about how government agencies can become successful by partnering with industry through public-private partnerships. And I'm going to illustrate that through two other stories. And one is about the current Ebola epidemic, and lastly about the H7N9 avian influenza outbreaks in China. And the reason I bring that up at the end is because that's a success story. The Ebola is still in transit, so we were still working on that. And to show you then, if you actually work hard on this and you bring, certain talents and partner with the right people, and that's always what it is you have to work with the right people, then you can actually make this happen. As you well know, that man-made and Mother Nature have dealt us blows over the past 14, 15 years, many of which we will never forget and have changed our lives irrevocably. So from 9/11 to Amerithrax to SARS in 2003 to H5N1 avian influenza reemerging in 2003 and 4, which that brought me from industry into government, to the H1N1 pandemic, in which we responded for my agency of making the vaccines that many of you, I hope, took, to the MERS coronavirus infections in the Middle East, to the H7N9 avian influenza outbreaks that killed a number of people in China, and then to finally what we see today with the Ebola, not outbreak, but epidemic. So we know that we have threats that are there. They're frequent, more frequent than we'd like to think, but we know that it takes certain things to happen to be able to address these. And there's some real facts there. Some of those facts are that drugs and vaccines are expensive to develop. They're a very risky proposition, and it takes a long time to do that. And in order for that to happen for an outbreak that is, you have to respond immediately, those are formidable challenges that you have to overcome. And you see up on that slide that you see a little triangle called the Valley of Death. For many of you that know the pharmaceutical industry, you know it very well. That's the area where you go from preclinical, the animal studies, to actually putting it into people to see if it is safe and it works. And most companies, including large pharmaceutical companies, have a real problem with that they cannot overcome that valley of death. Indeed, when we actually had to respond to anthrax or smallpox, pandemic influenza, how could we actually have companies go over this valley of death to rapidly be able to develop these products? We threw a lot of money at it in 2004 with an act called the Project Bioshield, in which we thought large pharmaceutical companies were going to come in, take the money, and then make anthrax vaccines and many other drugs and vaccines. It didn't happen. Did not happen. Only the small companies that were very inexperienced were willing to take a risk on that. So in 2006, Congress had to readjust our sights and the way we did, and that was when they had an act called the Pandemic and All Hazard Preparedness Act. It brought BARDA, the Biomedical Advanced Research and Development Authority, into existence. It's the federal agency that you've never heard of. In fact, if you were to Google us about 3 or 4 years ago, we were still below the superhero doll that you would see. Fortunately today, we've had more hits today, and so we actually outrank that doll. But hopefully what I can convince you of is that we've actually had more impact in a positive way than the comic book superhero doll. And part of that was actually bridging this valley of death, helping companies go forward for these types of drugs and vaccines and diagnostics that needed assistance to get across this. And money alone was not going to help. So what did you have to do? You actually had to have a agency with the federal government that has its only mission, its only thing it did day in and day out was to support that development of these products going forward. And secondly, and I think even more importantly, was that it actually had to be composed of individuals that came from industry, from regulatory agencies that had actually done this kind of work before, knew what failures, what traps were there, and could move this forward. Well, I put that also up there, a little square that says the Ebola medical countermeasures. You see where that is? It's at the bridge between going from non-clinical or preclinical work into phase 1 human trials. That's where we were August of 2014. Not at the end where you have licensed products, but that point. And look how long it takes you to get there normally. From that point on, 8 to 10 years if you're lucky, and at least $1 to $1.2 billion. And we were faced with a situation that we had to respond immediately, come up with and take those candidates, at least we had some, and be able to compress that not by a few months, but take that by years, from 8 to 10 years down to months. And so that leads me into who was going to do this. Well, there were 5 agencies in the U.S. government that were responsible for doing this. One was the CDC, that the surveillance, being able to do control intervention in the affected countries in West Africa. Certainly NIH by investing in the early development and taking preclinical work. Some of the DoD agencies like DITRA and DARPA with some of the early discovery work. And the FDA with a regulatory role. But could there be an agency that could actually help bring those candidates through from early development right in through advanced development and hopefully actually at the end of the day have licensed products? Well, there is an agency, and that's called BARDA. And our mission is to support advanced development and to make available those drugs, vaccines, diagnostics, and medical devices that would help for bioterrorism events such as chemical, radiological, nuclear, and biothreats, pandemic influenza, and emerging infectious diseases. And we do it through innovation, through support of the development, the building manufacturing infrastructure, and also really helping the companies with core service assistance. And I'll explain that a little bit more later. This is all possible because I'm the very humble and very fortunate leader of 157 scientists that we have in BARDA that are dedicated for this proposition, and they didn't come here to be in the newspaper, they don't come here to get rich, and they don't come here to make their bones. Many of them are in their second retirement, and they've come here to help the country, and in this case the world, with these types of what we call medical countermeasures. Since we took over in 2007, when BARDA was created, we have had 160 different product candidates through our pipeline. And so you can see through the different events and the different years as we moved forward, we've built that pipeline, and both for pandemic influenza and for what we call the bioterrorism events. What you don't see there are emerging infectious diseases until 2014. So we went from 0 to having as many as 8 different candidates in a matter of months in 2014 and today. If you actually put that same time scale against industry with a pipeline like that, we would be ranked about number 8 in the world. We've invested about $15 billion over this period of time, about $1.5 billion in R&D every year for advanced development. Not discovery, not early development, but advanced development, which is the most difficult part, in fact, and the part that costs the most. So, tell me, have you done anything though? You've got all those candidates there. The answer is yes. Under Project Bioshield, we've actually delivered 12 different new products into the Strategic National Stockpile, or at the manufacturers are available if we have an anthrax attack, a smallpox attack, if you have botulism as a terrorism weapon, ionizing irradiation from an improvised nuclear device, or from a 10 kiloton nuclear event, and also from chemical agents. Didn't exist before. Before BARDA was around, these did not exist, from anthrax antitoxins and vaccines to botulism antitoxins and anti-neutropenia cytokines, which were already on the market for hemonc indications, but they weren't for radiation. And certainly another midazolam, which is an anticonvulsant, can be used now as a much better drug for chemical agents. Well, how many have actually been approved by the FDA? Since we began, we've had over 20 drugs and vaccines, and medical devices, and diagnostics have been approved by the FDA. In the last 2.5 years, 8. And here are just a few of those from influenza vaccines, whether they be cell-based or recombinant-based, modernizing influenza vaccines, to pandemic vaccines, again, next generation portable ventilators, and diagnostics for not only influenza, but respiratory syncytial virus. So just a few there. Now, back to Ebola. Why are we there? For 2 reasons. It's a biothreat, and the Department of Homeland Security has shown that it can be weaponized, and that therefore we, as safeguarding the civilian population of this country, we actually are responsible for making sure there are candidate products there. But it's an infectious disease. And there have been 25 outbreaks before this past year, and they were all outbreaks. Why did it change so much? What happened that we went from an outbreak to actually having an epidemic? Well, one reason is that it went from rural settings to urban settings. And no one had any modeling data or any real thoughts about what would happen if these viral hemorrhagic fever viruses moved into an urban setting. Well, it also went into areas that were war-torn. Their health care infrastructure was marginal at best. And this was an area in which we had not done a tremendous amount of work from NIH or DoD in actually having clinical networks, study networks and research and development frameworks set up already. So this is Liberia, Guinea, and Sierra Leone. So what we have today is about 20,000 people have been infected. Only about 10,000 have died, unfortunately. And we actually had modeling from the CDC that said by the end of December or early January, we'd have about 1.2 million cases if we didn't do anything. Well, we did. CDC and the World Health Organization and many other agencies like the MSF, the Wellcome Trust, and many other, the Gates Foundation and other NGOs went in and they changed the culture, and the other companies went in so that the normal epidemiological interventions started working. For example, if you change the way that individuals that are infected and that die, you handle the bodies, you can have a huge impact on the individuals that are infected afterwards. There are actual case history studies in which individuals had died. Body fluids were then transferred to loved ones that actually visited the body, and they became infected. If you change that just one thing, it has an impact. Well, imagine doing that across the board with many other things. And we understood that you didn't have to die. You could have health care to a point in which you didn't have to die, except for one person in the United States that was evacuated to the United States. Mr. Duncan unfortunately passed away. Everyone else survived. So we know that you can take that 70 to 80% mortality rate and you can bring it down very, very low by just really good health care. Well, we also thought at that time with the 1 to 2 million people maybe being infected and then half of them dying, that we had better take heroic measures. And I will say this is one of the times I've seen industry, non-governmental agencies, and government agencies and nations across the world unite to work together on this. And this has been very sobering because we've learned how we have to interact, because we haven't had to interact in this way before, and it had to be done now. So one of the things that we became involved in is developing the vaccines and some of the therapeutics for this. And we brought our core principles for BARDA into this space. First, we exist to bring these to address the medical consequences of biothreats and emerging infectious disease. That's easy. Work with other federal partners. We've done this before, especially with pandemic influenza, with CDC, FDA, NIH. We've done that routinely, and with very great success. But to also include our partners over at DoD. Now, why is that? Why even bring that up? They have been invested in this for the warfighter for a number of years. But not every country around the world welcomes our defense comrades there. And that was a political issue that we had to overcome. Why are we there? Did we bring the virus there? Is this some problem that the defense departments in their countries are going to have to deal with ours? Fortunately, we've worked through that. And I think that we have, in all three countries, a good understanding and diplomatic relations that have much improved, so that when we leave, we won't be leaving them empty-handed. They will have a better healthcare infrastructure. They will actually have scientific and research frameworks and networks that they work with NIH and BARDA, CDC on a regular basis, that we can help them not only with this disease, but with others going forward. The other part is that something we do, I think we do very, very well, and it's usually when you hear with government working with industry, it's usually, oh, it's a disaster. Fortunately, I think we have been able to show that model is not correct all the time. And that is that you can make public-private partnerships with industry and with academia, and to actually be able to develop many of these types of drugs and vaccines. And you can do it when you have to have it there immediately. And even after doing this for a number of years with the federal government, and a number of examples, I'm still astonished how well we did. And I'll tell you a little story in a moment about how we were able to work with some of the major biopharmaceutical companies for monoclonal antibodies, and they responded unbelievably well. So those public-private partnerships were part of it. The other part is that we actually need to have an infrastructure that really didn't exist before to be able to respond. We needed to have it to help developers. And I told you that earlier, the valley of death. But you also have to have it to be able to do things in a response mode. You got to do non-clinical studies. You got to test in animals. Secondly, you've got to be able to develop and manufacture these products. Third is you've got to be able to test them in clinical studies. And fifth, you've got to be able to put them into vials so that you actually can get the product there. And that turns out to be a real issue. So that infrastructure is there every day, but it's also there for an emergency. And lastly, you cannot be able to respond effectively if you can only do it at lab scale or at pilot scale. You've got to do it at large commercial scale. And therefore, you have to have the infrastructure with companies to be able to do that. So this is the landscape, the pipeline of Ebola vaccines that are in development. And what you see is that there are some candidates that are in what we call phase 2. That means that they're going into clinics in West Africa right now and starting to look whether or not they are safe and they work. They're what we call efficacious. 2 of those with GlaxoSmithKline and the NIH developed. We call it the ChAd-A3 Ebola vaccine. Another one is a small company called New Link Genetics that partnered with Merck, and that's using a different vector. We call it the Sackler stomatitis virus. For those of you that know cattle, that's a virus that can cause them some problems, but it actually works for making a vaccine pretty well. And what you see is in the purple boxes, those indicates ones that we, BARDA, have supported. So everyone is doing it, other agencies are doing part of it. In this case, the FDA approves the regulatory authorities to go forward with this. Secondly, NIH is conducting the clinical studies in the US and also now in West Africa. They started the clinical trial with these 2 vaccines last Monday. The trial is enrolling, and they will enroll about 27,000 persons over the next several weeks and months going forward to show whether or not these vaccines work. And then BARDA is actually working on the manufacturing. And we're working with the companies to make sure that they can actually make this not at pilot scale, not the tens of thousands of doses of vaccine, but the millions of doses of vaccine. So the moment that the vaccine has been shown to be safe and efficacious, or that it works, that they can actually make the millions of doses of vaccine later in 2015 and have a mass vaccination campaigns if that is what's needed at that time. Other vaccine candidates that are out there are with Johnson & Johnson and Bavarian Nordic, both of which we're working with, and we're in negotiations with them right now. Another one is from Novavax. I know them very well. That's where I actually was in my last industry post, where I was director of vaccines there, and they have a Ebola vaccine. Both of those are in phase 1 studies now. And so if those look good, then they'll move into efficacy studies going forward. Another one we've actually invested in is one Infectious, which Wyeth used to work on. NIH is actually looking at a rabies vaccine, in which they're using the rabies vaccine and putting the Ebola protein in there that would actually be able to use for rabies and Ebola. And so again, that's in preclinical development, but that gives you the landscape. So what you will hear over this month, clinical studies have started for Ebola vaccines this week in Liberia. They'll start next month in Sierra Leone, and CDC will be running that clinical trial with BARDA, and we're there using what we call our clinical studies network to actually help them on the ground, and our staff will be on the ground to actually conduct those clinical studies. And then the Johnson & Johnson vaccine will probably be going into Guinea in the coming weeks also. What about therapeutics? What about those drugs that actually can resolve the infection in people that are really about to die? So again, what you see is the same story, early development. There are a number of candidates that were out there, and I'll walk through some of those. BARDA made a big investment into what we call the monoclonal antibodies because they were very specific for this virus, and they were shown in non-human primates to protect all the animals that were infected with Ebola 4 and 5 days after they were infected. And that's very important because you may not show symptoms at the first day. In fact, you don't show symptoms. You want to be able to actually have a drug that will work when people actually know that they're really getting sick. And that's sometimes a problem with some of the small molecule drugs, in fact. So we hedged our bets, though, because the only monoclonal antibody which was discovered at the January 2014 that worked against Ebola, at least in the animal models, was one that was a cocktail from Canada and from the United States. And it grows in tobacco plants. So R.J. Reynolds actually has a plant in Kentucky that actually makes this product, and it had only been made for animal studies. It had never been made for humans. So that was the hand we were dealt. We said, you don't usually make monoclonal antibodies in tobacco plants. You make them in mammalian cells, and what we call CHO cells. There are great products out there that have saved people's lives with cancer and with rheumatoid arthritis and many other diseases using that technology. And also the fact that you can make it at commercial scale in very, very large amounts, in fact. And there are really fully integrated pharmaceutical companies that do this. But there's one problem, and it was actually shown by the scientists in Canada, that if you take the tobacco version of this monoclonal antibody and what we call the CHO cell version, the mammalian cell version, the tobacco one is much better. It saves 100% of the animals' lives, and the tobacco was only about 50%. So something was very different about that. And what was discovered is that these monoclonal antibodies that actually bind to the virus and actually neutralize it, that if they had sugars on them, specific sugars, that they didn't work so well. It actually inhibited their ability to bind to the virus and to neutralize it. So what do you do? Well, you can put them in tobacco plants. The tobacco plants are all in pilot facilities, so you can't make a large amount of it. And we said, we're going to do that. We're going to do that, and we're going to get some other tobacco biopharmaceutical companies to make it too, and we're doing that too. But we have to take and start from scratch and say, can we actually get some of the companies that make these monoclonal antibodies in CHO cells to find out a cell line that actually doesn't put sugars on? We went to the Amgens, the Lonzas, the Regenerons, the Genentechs of the world, and said, what do you think? Can you help us? And so they talked to us, we looked through it, and we could remove the sugars, we could genetically have them such that they're not there. And what we chose to go the genetic way. And so Genentech and Regeneron came through for us. They started from scratch. They said, we will take the sequences for these monoclonal antibodies, we will put them into a specialized CHO cell line, and we will make these not at phase 1, but going all the way back to discovery and take them through, starting back in last September. And what I can tell you today is that they're now entering the non-human primate studies to see, and we'll go see whether they work or not. So in 5 months, they have gone from discovery all the way to being close to being to phase 1. If they work this month in the non-human primate studies, then we will move forward into clinical studies immediately in March. They already can make these at commercial scale because that's what they do. And then we'll have a much larger supply of those antibodies and be able to hopefully have that not only for this epidemic, but in the future as we go forward. What did that tell me? That told me that if we work with the companies, put a little competitive edge in there because they knew they were working against each other, that they can take with their normal process and take it from 2.5 years down into months, provided the government gives them the impetus and the urgency to do it. And they're great partners. I have to say, fantastic partners on both sides. So going forward, there are some other drugs. Now, this is called ZMapp. This is one you've called the secret serum. It's been given to 6 individuals around the world that have been evacuated from there, and also 3 in Liberia, but under compassionate use. So we don't know whether it worked or not. So that's why we're doing the clinical studies at this point in West Africa. And that clinical study with ZMapp, the tobacco-based product, starts next week. We're shipping it on Friday. It'll be there Monday, and the studies start later in the week. So hopefully we will know very quickly by March whether or not that particular monoclonal works. Well, are there other products out there? Well, there were several that were developed for Ebola and other viral hemorrhagic fever viruses. Some you've actually heard of. One is a molecule that is very specific for the Ebola viruses, the siRNAs. These are inhibitory RNAs. And unfortunately, if the virus changes at all, then it doesn't work so well. Well, the virus had changed from the older strains to what we have called the Guinea strain now. And so it had a couple of problems, and now they've made a new one. It doesn't come without some problems, though. And so that's a product called the Tegmira product from Canada. Another one that you've probably heard of is favipiravir, and that is used, it's in clinical studies for influenza. And we've actually studied this ourselves, and the Department of Defense is invested in it. This is an antiviral drug, and they thought that they saw some activity in the tissue culture plate, and then they moved forward with it. And that has been tested in Guinea, and unfortunately the committee there that looks over the safety of that clinical trial saw that it was not a good product and didn't really extend the lives of the individuals that were infected. So that study was halted. There is still some ideas that we may be able to resume that study in a different way, and that may actually happen. Another drug that you've probably heard is brincidofovir. Probably many of you heard in the news of this drug, not for what we were actually invested in, for smallpox, which it seemed to do very well there, but actually the little 11-year-old boy that was in Virginia that had an adenovirus infection, and this drug was, the company withheld it for about 10 days until the FDA and BARDA talked him into actually providing a clinical trial, and that young man is alive today, and he lives very well because he was able to do that, and also 50 other people now that have had that drug. But it was this drug called brincidofovir was never shown to actually work in animals, so it was just basically they thought they got a real good hit or a target in tissue culture and moved forward. That study was done in Sierra Leone by the Oxford University in the UK, and it has also been stopped, again for adverse events. As we've gone forward, what you're seeing is that those candidates that you have, they're winnowing themselves out. And so the most promising one we have right now are these monoclonal antibodies. And that's why we've put so much investment and so much time into that, not only into tobacco-based monoclonal antibodies, but also the new CHO cell-based ones. So we'll keep our fingers crossed. We hope it works, and we hope we can save some lives with that. But that's part of our process, because many of us came from industries that you don't bet on one thing. You have to have a strategy that actually looks at a number of different ways to solve the same problem. And certainly, hopefully, it will pay off as we go forward. The other part of this is you have to have the public-private partnerships. These are just two ones we've had not only for Ebola, but in this case for influenza and Ebola. One in Sanofi Pasteur in Swiftwater, Pennsylvania. The other in Holly Springs, North Carolina with Novartis. The other part is that you have to bring real technical services to this. That used to be a BARDA in the middle, but where the black box is. But our Ebola vaccines and our Ebola therapeutic candidates have actually been assisted by a number of different core service assistance programs that we set up over the past 4 years. First, we set up a non-clinical services network that has 17 different laboratories across the world that can do these these high containment studies with these really infectious and high mortality agents. And they have responded very admirably. They're doing the non-human primate studies as we go forward. Secondly, we had we call Centers for Innovation and Advanced Development and Manufacturing. These are flexible, rapid response manufacturing facilities that are a consortia of academia and industry with us. There are 3, one in Texas, one in North Carolina, and one in Maryland. The third is with Fill Finish Manufacturing Network. These are the people that actually, in a sterile environment, put the product into the vials. Very, very important. It's done right. And it's one area in the pharmaceutical industry that when things go bad, that's usually one of the areas that is located that's a problem. And so we have 4 contract manufacturing organizations from across the United States that do this. They not only do it for these Ebola candidates, but they also do it for drug shortage products and also for pandemic influenza if we have another pandemic. And lastly, our clinical studies network, which is only set up last year in February of last year. Wow, we were just trying to get them started, and we threw them into the deep end late last year, and we said, you've got to respond. You've got to set up a response effort. And we are working with CDC, and we have 5 clinical research organizations from the United States, and 2 of those are actually going to be with us in Sierra Leone, actually boots on the ground conducting the clinical studies and doing the data management and analysis of results of that as we go through our vaccine clinical trial with CDC in Sierra Leone. Had we not had these, we wouldn't be playing a very vital role in this response or other responses. So you have to have that. This just shows you geographically where these Centers for Innovation and Advanced Development Manufacturing, they'd be making monoclonal antibodies for Ebola. And then these are the fill finish manufacturing organizations that we're using. They filled ZMAP. The ZMAP that's being shipped over on Friday, they did that up in Bloomington, Indiana with Baxter. And you have to be able to make these products at commercial scale. And what you see on the left is the Novartis facility, they're making monoclonal antibodies, and then the Merck facility making the vaccine, one of the vaccines that's going forward right now in West Africa. Bottom line is that BARDA Ebola response is global, and it's across the world with different partners, from Canada to Europe to our West African colleagues, and moving forward here in the United States. Now, the last story I want to tell you about is influenza pandemics. And it was very well said, 1918 was a turning point in socioeconomic and certainly the world's military history, because it changed the way that World War I happened and the results of that. And certainly the way that we approached it. In 2003, we thought we were on the brink of another severe H1N1 pandemic like we saw. A number of books were written. The previous administration really believed in that we need to be prepared for this. They actually asked me to come out of industry, and they nearly broke my arm to do it. And I really thought I was going to come out of industry and set a pandemic influenza program up for 2 years, and I'd go back into the industry. As one of your members had said, once that public service bug bites you, be careful, because when it does, it never leaves. And so that is what happened. And so I've been there almost 11 years now. And but I went there for pandemic influenza to set up a program so that we can make new vaccines, new drugs, some diagnostics, make some new factories, and then I was going to go leave. But lo and behold, we had some other things happen. We had the 2009 H1N1 pandemic. We had outbreaks just at state fairs in the Midwest with we call it the variant H3N2 virus. That a number of children were sick. if you remember in 2012 and 13. And then in China, we had the H7N9 virus appear. Very pathogenic, caused high mortality, but it didn't cause a disease in the birds, which made it extremely difficult to diagnose just by looking at a flock of birds in the poultry markets. Not like H5N1, which also was very fatal for humans, but that infected birds, they got sick and they died. So now you had a virus that you couldn't pinpoint exactly which birds had it, so you had to call all the flocks. Hundreds of millions or even billions of birds were called in 2013. The virus still persisted. And why were we so worried about this particular virus? Two reasons. First, if you look at the genetic sequences of this virus, you saw that it already had made a mutation that would allow it to be transmitted from birds to man much easier than some of the other avian influenza viruses. And all we needed was one more change. And if that happened, then it would be a pandemic of enormous explosion. A virus not like the H1N1 of 1918 that killed about 2%. This would kill 50 to 70%. This would be a plague that we have never seen on this planet. That we were very concerned. And of equal concern was that we had been trying to make vaccines against H7 avian influenza viruses for about 8 or 9 years. We couldn't do it. I mean, we had really tried hard. Academic, industry, and government labs together working on this. But we had a few more tools now than we had before. And so what we went forward with from these viruses is we first did a risk assessment, and we said, how bad is this? And when we did the risk assessment, looking at the impact versus the risk, and this really looks at how quickly is it transmitted and how pathogenic is it? How much disease is it going to cause? It was right on top of H5N1. It was right on top of 1918. So we said, yeah, we have to do something. Fortunately, what we had in our toolbox now were things called adjuvants. Things that actually, these substances that actually could broaden the immunity and give you much greater immunity. And secondly, we had new ways in which we could actually make the vaccines. The way we've always done it for influenza virus vaccines for the past 65 years, there's a virus, we isolate it from clinical samples, we take that, we grow it in eggs, and we move it forward, and then when it grows really well in eggs, because most of the time when you first get them, they kill the eggs, and then be able to make it into a high production to make those. Well, Craig Venter's lab out in San Diego, who was part of the people that actually sequenced the human genome with in competition with Francis Collins, and Novartis, working with BARDA, actually came up with biosynthetic technology. So that on April Fool's Day, 2013, we got the sequence from the laboratories at the CDC in China, which our CDC had actually helped set up. We saw the sequence all together, and we were all on the phone at the same time. And when we looked at this, we said, we don't need the virus. You can keep the virus. All we need is the sequence. From there, we actually made the virus vaccine. And it was done not in months, but in days, 7 to 10 days. From there, we went on, and we, as you see in this diagram, we made the vaccine seed preps. We went into clinical lot manufacturing, and we were in the clinic in July. Fastest we've ever done it. We then moved right into clinical trials in August, and by September, we knew we had a vaccine, and it worked. It was safe, it was immunogenic, and we then dodged a bullet. The epidemic or the outbreaks in China started to wane. We had vaccines, and we had actually 5 vaccines that actually worked. And so we said, what if it comes back? Well, we better make some stockpiles of this. So we have enough vaccine stockpiles of H7N9 for at least 40 to 80 million people right now. And we keep those at the manufacturers, and we're ready to go. That's the fastest that we've ever been able to do this. If we compare it to what we did in H1N1 back in 2009, where it took about 23 weeks from the time we saw the first virus in May to the time we actually had the first doses available, which were in early to mid-October, it's 23 weeks. In about 15 weeks, we had this. Why? Because recombinant technology and the efforts that we had been going forward with modern technologies to make these vaccines. So those investments that we made already were starting to pay off. And so this is basically the results of that. We actually did some very esoteric things. Unless people care about vaccine manufacture, it doesn't matter. But things like we call potency assay, actually trying to understand how strong or what's the strength of the vaccine. That takes a long time to do. If we could actually do that in a couple of weeks, we shave time off. And what we did with all these different ways is we shaved times off. And why is that important? We learned this from H1N1 2009 pandemic. The sooner that we can actually have vaccine available, the more lives that we can save. If we can actually have the vaccine there before the peak of the epidemic or the pandemic, the more lives that we save. And so that has been a burning passion of my folks, people at CDC, FDA, and NIH. It's how can we shorten that time clock? How can we actually do that? And working with the manufacturers, we're actually knocking weeks and weeks off of that. 23 weeks to 15 weeks. That's a big improvement. Because if you get 10 to 20 million doses of vaccine out every week, that's more people every week that are being saved. One of the stories, in addition to building domestic manufacturing capability, where we were at 50 million doses to now being able to make pandemic vaccine capacity at 600 million in the United States, is an investment we made for $65 million partnering with the WHO in 11 countries in the developing world. They had 0 doses of influenza vaccine in 2005. In 2004, I went to Vietnam and was working with the Vietnam government and a company there trying to help them make a vaccine. And we said, this is not going to work this way. We have to go out and reach out to the developing world and teach them how to make influenza vaccines. Help them build their facilities. Help them, teach them how to actually make these vaccines. And then once we do that, go back there and make sure they know how to do it. And so using those principles, we've actually been able now to have 4 of those vaccines licensed in those countries from all over the world. You see them here. To having a capacity of 0 to today, 337 million doses of pandemic influenza vaccine capacity. By the end of this year, we'll be at 500 million. That's not enough, but it's certainly better than 0. And it just says that for every dollar that we put in, we got a return of investment from the governments or other entities of anywhere from $6 to $17, depending on the country. Great investment. Again, that's partnership around the world. Now, I want to leave you with some hope, okay? And so I testified before Congress with Dr. Tony Fauci, Dr. Ann Schuchat, and Dr. Karen Mithun last Tuesday. And we were raked over the coals because of 2 things. One, measles, and why people aren't having their children vaccinated for measles. We were all asked under oath, would you recommend that your child be vaccinated with a measles vaccine? All 4 of us said absolutely. And would you actually have your child vaccinated for influenza? And we said absolutely. It's a safe vaccine, and it does provide, even in a bad season where you don't have good match, some immunity there. But what Tony Fauci and I were brought there was to provide hope. What does the future look like? The last 65 years, it's been looked at, could we have a vaccine that you take for influenza and you take it one time in your life, or maybe only a few times in your life, instead of every year? And until 2011, people had been trying desperately to do this and failed. But it wasn't because they didn't put money into it and didn't try, and the best and brightest minds were on it. There was a discovery, and that discovery looks like this. This molecule you see up here, it's called a hemagglutinin. This is actually on the outside of the virus. It actually binds to red blood cells, and that's an easy way to actually assay it. But more importantly, these are the proteins that actually bind to your lungs and your upper respiratory tract, bind there. So, and it changes the top of it, called the head, it actually changes very much because of antibody pressure around the world, and also going into animals and back to man, and back from man back to birds and swine. So it changes all the time. And therefore, we actually have to keep immunizing. But what if we could actually see if there were other places on that same protein molecule that actually could provide immunity to it? Two investigators, same year, made the same discovery. This region at the bottom, the stalk region, okay? Think of this as a tree or a broccoli stem, and the top of it being the head, and the stalk region down and below. They found that there were regions on that portion of the protein that every influenza virus had, no matter if it was H1N1, H3N2, or H5N1, or H7N9 virus. They were the same, and that the antibodies that recognized those regions would neutralize and kill the virus regardless. Whereas an antibody against the head, you had to be very specific. The ones for H1N1 would only work for H1N1. So that discovery then fueled the effort that NIH and BARDA have gone forward with, and that is to develop what we call a universal influenza vaccine. That's the holy grail of influenza. If we can do that, then we'll be in the history books. If we don't, then we're in a long line of others that have tried it. But we think this is going to work. And the reason we think it's going to work is because your body actually sees this portion, and we've put it in animals, and we've done phase 1 studies with this, and they actually look very promising. So now the NIH and BARDA, working together with the industry, are going to do a very complicated phase 1 study to actually see if it actually works against the human influenza viruses like H1N1, H3N2, against H5N1, H7, the H8s, the different influenza viruses. What we will accept is that at the least they will give us a more effective influenza vaccine. So the influenza vaccine you take next year or the next year will at least be better than what we've seen today with a poor match. So we won't see that. That they will recognize many different viruses. So the virus does change. If it changes up in this head region a little bit, it doesn't matter. You're still protected. And that immunity is long-lived. It's not just 1 year. That maybe it's 5 years, maybe it's 10, maybe it's even a lifetime. And lastly, and this is where BARDA really is in our scope of work, is that it actually can serve as a primer for a pandemic. You take this vaccine with these, what we call a chimeric HA vaccine. You take that, you take 1 dose of that, and when a pandemic occurs, you only need 1 dose of the pandemic vaccine. You already have some immunity to begin with. So you're primed already. That would be enormous if we had a severe pandemic. It changes the paradigm, it transforms the way that we actually prepare for a pandemic. And so that's the hope I want to leave you with. The other part of it is, well, those antibodies that recognize that, can't we use those for a therapeutic? Yeah, because those antiviral drugs you take, the Tamiflu, Relenza's, even the ones that we've supported and invested in, Peramivir, which is a single dose antiviral drug that works intravenously for severe cases of influenza in hospitals. These antibodies actually work against all those viruses, and you don't have to worry about drug resistance. And that's a big improvement if we can go do that. So I want to leave you with that hope, and hope that who you're going to call when the next big epidemic occurs, hopefully you'll be calling on BARDA. Thank you.
Thank you very much, and maybe we could take a few questions. Do we have time for just a few questions? Sir?
Hi, Ben Alzer with the TB Alliance. We're one of the groups working on sort of partnering across sectors to develop new therapeutics for tuberculosis. My question is just wondering how can we apply the successes with Ebola to other infectious diseases like drug-resistant tuberculosis, where we've clearly seen with Ebola that a lot can be done if the will is there. And when you have someone like BARDA coming together to organize the actors in the field, and I'd just be curious to get your take on how we might take that example and apply it elsewhere.
Very timely question, because BARDA has been interested in tuberculosis, especially drug-resistant tuberculosis, for a number of years. And we, by mandate, by law, we are supposed to be working on emerging infectious diseases. And clearly that falls in that category. Your colleagues have actually been to see us a couple of times. And one of the things that we're doing this year is that we're setting up a new division within BARDA called the Emerging Infectious Disease Division. It will do two things. It will prioritize 4 of 5 major emerging infectious diseases, and tuberculosis is on that list, in fact. And we will apply the same type of principles and work with the same diligence towards those, and also be able to respond to in an emergency to produce vaccines, therapeutics, diagnostics, where we don't know what the emerging infectious disease until maybe weeks or months later. So we have ways to do that. So the answer is yes. We're very interested, and we look forward as we go forward in our FY 2016 to include that in our budget as we go forward.
Tom McCardle, thank you for your time. You mentioned manufacturing and scaling, but we still don't appear to be at the point where we can change, for example, the annual flu shot to make up for a poor batch mid-season. How close are we to doing that?
So, great question, one that Congressman Murphy asked me also. So there are some really interesting studies that have been going on that we and the NIH have been supporting, in which you can use evolutionary biology techniques and go forward and predict what are the possible new strains going to be. And you can put selective pressure on them. Because it turns out the virus is only going to change so many ways. We now know that. It can't change in hundreds of thousands of ways, because the viruses won't grow. But there's only so many ways. And if we can predict those in a much better way, then we can actually then help inform the strain selection in the interim time. But more importantly, we can influence the design of the vaccines. And there's an interesting real concept that's out there. We're actually about to do clinical studies with this. It's called the back boost phenomenon. And what you see is that the vaccine you take today, and individuals that have been infected with influenza or they have been vaccinated for influenza, that vaccine is going to give you really good immune responses against last year's and the year before's circulating influenza strains. It's sort of intuitive, you kind of know this, but it actually does. If you could predict what's going to be there in the next 2-3 years and make the vaccine against those strains, you make those vaccines, it then gives you that same back boost phenomena, so you would make a really good response against the ones that are actually coming up, because the ones that are going forward all came from that would come from the same progenitor. So we're going to test to see if that idea works. And if it does, it may change the way that we actually select strains and, more importantly, make vaccines.
Time for one more question, sir.
Thank you very much, Dr. Robinson. Excellent presentation. I'm actually going to be deployed to West Africa next month for Ebola.
Thank you for your service.
To take a look at your presentation today. My question is, for the past couple of days, we have been hearing a lot of questions of the validity of vaccines from the leaders in our country. What is the political buy-in from West African countries in terms of starting clinical trials of vaccinations there?
Great question. First of all, that we were asked, what did the senior public health officials in this country think of that? And we always point to, and we did that in the hearing, vaccines and antibiotics are the two greatest medical miracles in mankind. They've eradicated diseases and allowed our world to actually grow to its population today. In West Africa and Liberia, I can say they are embracing it, and they are flocking to the clinical sites in Liberia. In Sierra Leone, that study is about to start. And an interesting thing, a cultural thing there, you can't say that they are Ebola vaccines. You have to say they are Ebola preventive vaccines. Because if you say Ebola vaccines, it's thought of, well, you're giving the virus. And so, part of it is communications and understanding. The other part is that we are helping them do the clinical studies. So let's make that very clear that they are in charge. Their regulatory authorities have to accept, their principal investigators have to accept what's going on and are in charge of what's going on. We help them with the logistics, we bring the vaccine to them, but they are in charge. I think by that kind of partnering where we're empowering everyone to do this and do it as a team, I think the acceptability. And from what we know in Guinea, the same thing is happening there too.
Well, thank you very much, Dr. Robinson.
Thank you. My pleasure.